Assalamualaikum warahmatullahi wabarakatuh. Welcome back in my class, Teknologi Pengendalian Hama. After we study about the implementation of integrated pest management, do you still remember what are the principle of IPM? Do you still remember? The first one is growing a healthy crop, and the second one is optimizing the use of natural enemies or biological agents such as predator, parasitoid, and entomopathogenic. And the third one is we doing monitoring. We observe daily and weekly in our field. And the fourth one is farmer as an expert. So in today's lectures, we will study more about how to optimizing the biological agents using predator and parasitoid. As you see in the pictures, what is the pictures? This is the egg of the hemipteran insect, telur dari hemiptera ya. Egg of hemipteran insect, which is pest, and this is parasitoid. The parasitoid is emerged from the egg of the pest. Yeah. So this is the parasitoid. We will study more about this biological agent in the next slide. And now we study about the biological control. What is the biological control? Biological control, PH ini adalah pengendalian hayati or biological agent. Activities of natural enemies that let the average population of the target organism is lower than if it had no control. As you see in the pictures, this is the economic threshold. So we using biological agent to minimizing the population of the pest. Like in the pictures, this is the green one is by the biological agent. The impact of biological agent. So we keep the pest population below the economic threshold, like in the picture. So by using biological agent or pengendalian hayati, we make sure the population is still below the population, the economic threshold. And please remember, in the integrated pest management, we cannot destroy all of the pests. Why? Because if we destroy all of the pests, so the impact is imbalanced ecosystem. So what we need is only we keeping the pests below the damaging level, keeping the pest which is still safe. Yeah. So our crop still fine because of that pest. Biological agent is means of keeping pests below damaging level through the activities of predator, parasitoid, and insect pathogens. You still remember that insect pathogen is organism that microorganism that cause illness in insect. Yeah, that microorganism could be fungi, could be bacteria, nematoda, protozoa, and viruses. So we can make the insect become ill because of that organism. Entomopathogenic fungi, entomopathogenic bacteria, entomopathogenic nematode, protozoa, and viruses. So this is the insect pathogen, and this is the definition of coolant and instance. Predator. What is the predator? Predator is an organism that eats more than one organism during its life. So the predator needs many organisms to fulfill their life. And the size of predator is usually larger than its prey. Yeah. And parasite, what is a parasite? Parasite is organism that live at the expense of another host and which does not usually kill. So parasite the host um, does not usually kill. It's different with parasitoid. In parasitoid, the host uh, will be killed by that biological agent parasitoid. Parasitoid is a parasite that kills its host, but not directly kill. There is a process to kill the host. And how about the size? The parasitoid usually smaller than host, usually smaller, but in some case, the parasitoid is bigger than its host. The next slide, there will be pictures. Yeah. Now we move to the definition of parasitoid and host. What is parasitoid? Parasitoid is an organism that lives on or in a host organism and ultimately kill the host. So this parasitoid is could be live on or in. Jadi bisa hidup di dalam atau pada inangnya ya. Organism that lives on or in the host and ultimately kill the host. And what is the host? Host is animal that attacked by the parasitoid. Yeah. Commonly, the host is from insect or other arthropod like spiders. And the most insect parasitoid found in the order of Hymenoptera and some in Diptera. But the highest parasitoid is come from the order of Hymenoptera. In many species, eggs are laid within the bodies. Yeah. In this case, the adult parasitoid is searching for the host, and after they find the host, they insert they will oviposit their egg, and they inject their egg into the body of the host, and then the egg become into the larvae, and the larvae will live inside or outside the host, for example like that. This is endoparasitoid, so the larva will remain in the inside body of the host, and after larvae and then become into the pupae. While the larvae become into the pupae, the larvae will come out from the host body, and then the pupae is like that. This is the pupae of the parasitoid, and after the pupae, the parasitoid will the adult parasitoid will emerge from the pupae. Okay, I hope you understand what I mean. The egg hatch and feed on the internal organ of the host. So the larvae of the parasitoid will eat the internal organ, but it's not directly kill the host, because the larvae of the parasitoid will eat only the non-essential organ. That's why the host of the parasitoid is still alive during the development of the parasitoid. Yeah, feed on non-essential organ, thus feeding without killing the host. So the host of the parasitoid is still alive. And then after that, larva feeds on the fetal organ, killing the host, and pupate within the host, and then emerge as an adult and killing the host. And as I told you before. Okay, this is the pupae of the pest, and then this is the parasitoid. That's it, so small. Most of the parasitoid is smaller than its host. Characteristic: parasitoid usually destroy the host during development, so they eat the internal organ and a non-essential organ. Yeah, That's why it's not directly killed, but the host is still alive during its development. The parasitoid host usually in the same taxonomic class. The host could be from Insecta or from arthropod. Commonly smaller than its host, adult parasitoids are free living. Uh, the adult parasitoid is not parasitizing, no. The, the only stage that parasitize the host is the larva stage or the imager stage. The imager stage of the parasitoid are parasitic. They can live inside the host in or outside the host. Generally, the hosts do not immediately die, and habitat differ between adult and pre-adult. As you know that the adult parasitoid is free living, dia hidup bebas, while the pre-adult or the major stage are parasitic, yeah, on the host. And then this is the plant defense against insect herbivore. What the plant can do while the crops or the plant is attacked by the pest, they do communicate. Yeah, the plant is communicate. How they can communicate? Yeah, the plant, for example, the leaf commonly they release the small quantities of volatile chemical or secondary compound. They release the secondary compound. What is the function of the secondary compound? The volatile compound will attract both parasitic and predatory insects. They will come to the plant and they will parasitize the pest. So this is what the plant do. They also communicate to attract the natural enemies to come. Yeah. And also, the natural enemies will come to the plant also from the cues of wounding. Wounding itu luka ya. The pest is the leaf is eaten. For example, the leaf is eaten by the pest, so they are wounding. Ada luka, and also from the chemical in the saliva of the pest, and also the volatile compound that released by the plant. So all of the cues will attract the natural enemies. Uh, Both parasitoid and predator will come to that plant. Okay, and what are the example of secondary plant compound or volatile compound? There are many. For example, terpenoid, alkanoid, ester, acid, alcohol, ketone, aldehyde, and other. This is the journals that you can read about plant volatiles as a defense against insect herbivore. So also the organism like plant also do communication, yeah, to attract the biological agent. The importance of biology parasitoid knowledge. What we can do to to enhance the population of natural enemies. We can produce and do mass rearing. Mass rearing itu adalah perbanyakan massal, ya. We can breed the parasitoid in the laboratory, and after that we can release the parasitoid into the ecosystem. And what the important things that we should do? We should know the life cycle of the parasitoid, and we should do the behavior of that insect. And before we can develop the parasitoid in the laboratory, so the first one, we also we should explore. We do exploration of the parasitoid in the field. Yeah, we can explore the parasitoid, so we can find that parasitoid, and then we bring that parasitoid into the from the field into the laboratory. So we should know how to handling the parasitoid from the field into the laboratory, and then the breeding mass of parasitoid, how to breeding, how to develop this parasitoid into the laboratory. That is the important thing. And after that, we can release the parasitoid into the ecosystem. And then this is a site of attack on host body. The first one is endoparasitoid, and the second one is ectoparasitoid. What is the differences? Endoparasitoid. Endo is inside, yeah. So parasitoid which complete their development within or inside their host body. So adult parasitoid will search the host, and then after they find the house, they will deposit their egg into that house. They inject their ovipositor. They inject the egg inside the host body, yeah, and then after that, the larva of the parasitoid will live inside the body of the host. Usually in the situation where the host exposed. So endoparasitoid commonly uh, do when the the host is exposed. Jadi inangnya itu terlihat. Ketika inangnya terlihat, so when the host is visible by the parasitoid, it's easier to it's easier uh, to deposit their egg directly into the host body. This is the example of endoparasitoid. Uh, most of the parasitoid is smaller than host and then the host, but this is the exception. This is the parasitoid, and this is the aphid. The parasitoid is much bigger than the aphid, so this is the exception. The example of endoparasitoid come from the Chelonos species, and this is ectoparasitoid. Ectoparasitoid is different with endoparasitoid. Endoparasitoid is inside the host body, while ectoparasite, the insect parasite which feed externally of the host body and complete their development externally on its host. So they feed externally, and also they complete their development externally too. And the ectoparasitoid usually in a situation where host lives within protected location. For example, ectoparasitoid, um, we can find ectoparasitoid when the pest is not visible by the parasitoid. For example, is stem borer. Stem borer is dia adalah penggerek batang. The stem borer, the habitat of stem borer is inside the stem of the plant, so the host will not visible by the parasitoid. So uh, what the parasitoid can do to parasitize that stem borer? They will deposit their egg not direct into that body of the pest, but they will deposit the egg near the habitat of the host body of the host, near the habitat. So after the egg emerge as adult, egg hatch into the larvae of the parasitoid will uh, will search the stem borer inside the stem. Gitu ya. Jadi dia akan meletakkan telur, si parasitoidnya itu akan meletakkan telur di sekitar habitat inang, and then after the egg is hatched, si telurnya itu berubah menjadi larva, the egg hatch into the larvae, and the larvae will search the host on the stem. Dia akan mencari inangnya di dalam batang, gitu ya. For example, leaf miner, pengorok daun, and other. The example is Elasmus euplectrus. Okay. And the host range of parasitoid. The host range is different. Is divided into two. The first one is generalized parasitoid, and the second one is specific. The generalized parasitoid mostly come from the Trichogramma, Brachymeria, and tachinid fly. Generalized parasitoid is a parasitoid that can attack many hosts. So the host is many. But the specific parasitoid, the host is only some of the pests. Okay, this is the example of host parasitoid. This is egg parasitoid. For example, this is Trichogramma, the egg parasitoid, and they can. This is the egg of the moth, the caterpillar moth egg. This is egg of the pest, and they parasitize this egg. And this is the egg that has been parasitized by the parasitoid. The color is different. The color is brown, maybe brown or black. And then they develop the larvae and the pupas develop in that house, and then after that the parasitoid will emerge into from the egg. So this is the egg parasitoid, yeah, parasitoid. They feel the parasitoid finish their life cycle in the stage of egg, ya. Pada fase si telur inangnya itu masih pada fase telur, dia bisa menyelesaikan siklus hidup. And this process only 4 until 5 days, so it's really short. This is also the egg parasitoid, Telenomus. This is the stadia of the host, tadi inang egg parasitoid. So they will only parasitize in egg phase. The parasitoid larva, they will parasitoid only in the larva phase. Parasitoid pupa is the same, only parasitize in a pupa phase. For example, Xanthopimpla brahmeria, parasitoid egg, for example, Trichogramma, parasitoid larvae, for example, Aphanteles, parasitoid egg larvae. So it's mean that that parasitoid can parasitize in both in egg phase and also in the larva phase. For example, Chelonus. So this is what is difference between parasitoid eggs, parasitoid larva, and for example, parasitoid imago. The parasitoid imago is mostly from the dipteran insect, yeah, the parasitoid that come from diptera. The adult of the host still can be parasitizing by other insect from diptera. This is the life cycle of the egg parasitoid. The egg could be like this, yeah. The egg not only in round shape, but the egg could be like this. This is the egg of the larva of the pest, and this is the parasitoid. The parasitoid inject their egg into this egg of the pest, and then the egg of the parasitoid will hatch into the larvae. The larvae will eat inside the egg, and and then the larvae become to the pupae, and then they finish their life cycle. They will emerge from that egg too. This is how life cycle of the egg parasitoid. And this is different with larvae parasitoid. The adult will deposit their egg into the larvae, and then the egg of the parasitoid will hatch into the larvae. They will eat the non-essential organ. That's why the host will not eventually directly kill. Yeah, and before and then the larvae move change into the pupae. The pupae after the last instar, they will come. The larva of the parasite will come out from the body, and then they will the pupa will outside the host, and the pupa become change into the adult, and it's ready to reproduction again. They will copulate, they will mating, and after they mating, they will search for the insect to deposit egg. And then this is how the life cycle of larvae parasitoid. And numbers of immature per individual host. There is solitary parasitoid. Solitary parasitoid is a parasitoid that generally produce only one offspring per host. Jadi hanya satu dalam satu inang itu yang terparasit hanya satu ya. For example, from this one egg, this is egg of the pest, will come out only one parasitoid. This is called by solitary parasitoid. It's different with a gregarious parasitoid. In a gregarious parasitoid, in one host, yeah, we can find multiple eggs per host. Yeah, and after egg, and then they will emerge. They will move into the larvae, and the larvae after that into the pupae, and this is the larvae of the late instar of the parasitoid. And from one host, they will emerge many parasitoid that will come out from the host body. This is called gregarious. And how the process of parasitoid of getting the host? The first one is host habitat finding. From what? The parasitoid will easily find the host by the shape of the host and the feces of or dirt of the host. So the feces will also attract the natural enemies to come. Also, chemical coating on the host. The host, there is a chemical coating on the host, also from saliva, and also from volatile compound that produced by the host plant. So all of these cues will attract the habitat to come. And the second one is host habitat finding. Larva search and adult search. We can study more in the next slide. And after we find the host habitat, and then host finding, and then host acceptance, and the last one is host suitability. Yeah. So after they finish all of this process, they can develop greatly in the host. This is two different parasitoid host finding strategies. The first one is adult search, and the second one is larva host searching. What is the differences? Adult search is commonly endoparasitoid. So the adult parasitoid will directly inject their egg into the larvae of the body. Yeah, like in the picture, this is adult search. This adult can easily find the host yeah, they easily can find the host. But the differences between larva host searching, the host is not visible by the parasitoid. For example, like I told you before, like stem borer. Stem borer is a penggerak batang. The stem borer is live inside the stem of the plant, so it's not visible for the parasitoid. So it's hard to parasitoid to find the host. That's why in the larva host searching finding strategies, the adult parasitoid will deposit their egg near the habitat of the host. So after the egg hatch into the larvae of the parasitoid will go to inside the stem to find the host. So that is the differences between adult search and the larva host searching. And this is one of the case is how to controlling yellow stem borer. Serpopaca insatulas using Trichogramma sp. Trichogramma is a parasitoid, and this is a yellow stem borer, main pest of the rice field. These larvae of the pest live and eat inside the stem. That's why it's not visible by the parasitoid. Yeah. The symptoms of the Serpopaca incertulas are found before the rice flower known as sundep. So the symptoms could be sundep, could be beluk. Sundep is a symptoms that are found before the rice flower, sebelum pembungaan. And beluk, the symptoms of an attack is carried out after the panicle come out. Yeah. Beluk is a symptoms of the stem borer attack that can be found in a generative phase when the panicle come out, ketika fase generative fase malai. What is the differences between sundep and beluk? The sundep, the shoot of the rice stalks become dry, yellowish, and easily to pull out. Ya, rice stalk itu adalah pangkal batang dari padi, itu akan kering, menguning, dan easily to pull out itu gampang tercabut. Yeah, it's because the stem borer is they eat inside the stem. They eat the stem, so the stem will easily break out, easily pull out. And beluk, the attack occurs in the generative phase, causes the grain become hollow. The grain itu adalah hampa ya, hampa hollow. And how to managing this pest? We should implement the integrated pest management from the beginning, from the use of the varieties. How to we planting in unison. Planting in unison itu adalah tanam serempak or planting simultaneously in a large planting area, so that rice plant in the same pace. While we do planting in unison, tanam serempak, in the same time, so it will suppress the population of the pest. What will happen if we are not planting in the same time? So some plot in a generative phase, some plot in a seedling phase, some plot in a vegetative phase. So the pest will be easily moved from one plot into another plot. And we can, in integrated pest management, we can do mechanical control. Mechanical control, we can collecting egg mass of this pest. This is egg mass of this pest. We can easily remove from the plant and we can destroy that egg. And in this egg mass, maybe if it emerge, it will produce 100 or maybe 200 larvae. This is how the importance of monitoring of the pest. So we can monitor what's going on in our field. We can use insecticide application, systemic insecticide. Systemic insecticide is insecticide. Uh, it's different with the contact insecticide because this pest is live inside the stem, uh, stem borer, live inside the stem of the rice plant. So it's couldn't reach by the contact insecticide. The contact insecticide is hard to get that pest, so we can use systemic insecticide. So this insecticide, uh, the systemic insecticide are absorbed and transported through the plant. So that insecticide will absorb and transport through the plant. So it's different with contact insecticide. Utilization of natural enemies, both parasitoid and predator, also from entomopathogen. Now, this is the important thing. We can use Trichogramma sp, the parasitoid, and how to develop this Trichogramma by ourselves. We can study in the next slide. And this is the light trap. The light trap is the light trap is designed to catch the adult of this pest. Yeah, they will attract this moth, and they will trap into this light trap. And after that, we can minimizing the population of that pest in the field. And this is how to develop the Trichogramma sp by ourselves. This is was done by the farmers, one of the farmers in one of the group farmers in the Karawang, mass rearing. How we do rearing this parasitoid? We should use the alternative host because the life of the parasitoid will need the host which is pest. So we will alternate this pest into another alternative host. So we can reproduce more Trichogramma sp. We use Corsiracia palonica as the alternative host. This is the larva of the Corsiracia palonica was fed using the duck and maize and pork. And then because the Corsiracia palonica is also moth, we can copulate this adult of Corsiracia palonica into this breeding site. Yeah, after that we can get some of the egg of that Corsiracia palonica, because this is Trichogramma. Trichogramma is egg parasitoid, so we need more egg, yeah, to release that parasitoid. This is egg of the Corsiracia palonica in a paper. After that, we parasitizing this egg of the Corsiracia polonica by parasitoid Trichogramma sp. So Trichogramma spp will inject one by one this Corsiracia polonica. After that, we can release the parasitoid in the field. This is how the farmers do in Karawang. They also can improve the natural enemies, which is parasitoid, and they can do this. And this is how they release the parasitoid. They're using the plastic bottle like this. Yeah, this is to protect the parasitoid from rain. Yeah. This is there is a video that we can watch together. This is parasitoid, and this is the trichogramma. Trichogramma will parasitize the egg one by one. They only this is a solitary parasitoid. So from one egg will emerge only one parasitoid. So before they parasitize, they will inspect whether this egg is parasitizing or not. After that, they will deposit their egg in, one by one into that egg egg of the pest. Okay? And then after several days, it will parasitizing, and the new parasitoid will come out from this egg parasitoid, from this egg. Okay. This is interesting. This is the endoparasitoid, yeah, endoparasitoid. We will watch the full video, maybe in a Zoom. Yeah, this is how the lead instar of the larvae come out from the host body of the insect, and then they will pupate. They will apa, change into the pupae, ya. Pupae, and then the adult, and after that, after adult, the parasitoid will mating and reproduce by themselves and searching another host. And this is predator. Predator is an organism that eats more than one organism during its life. This is the example of predator. This is tomcat, Staphylininidae, Pyoderus fucipes. This is Hemiptera. This is apa ini? Mantodea. And what is the characteristic? Characteristic, mostly they are generalist. They can the prey is many. Yeah, they can eat many prey. Yeah. That's why this is generalist. Carnivore. They eat other animal. Heteroism. Heteroism is adult and immature insect life on the same habitat and a food source. For example, this one. This is ladybug. This is adult ladybug, and this is the larvae. Yeah, the larvae of the ladybug and the adult of the ladybug live in the same habitat. So sometimes from the immature stage and the mature stage is causing competition. Both of that insect is searching for the same prey. This is competition. And the fourth one is destroying or paralyzing prey before consuming. This is destroying before consuming. They destroy the pest before consuming. For example, like this, destroying, and some of the predator paralyzing before consuming. Dia memingsankan, paralyzing. And the predator generally require more than one prey for the development. It's really different. It's really different with parasitoid. Parasitoid only needs one host in each development, but the predator require more than one prey in the development. The body is generally greater and bigger than prey. The body organ well developed. Yeah, for example, in this Mantodiamantide, one of the organs that well developed is their leg. Their leg is adapted to catching and holding the prey. This is what is the leg in types? Raptorial, right? And this is Odonata, dragonfly. The eyes is really big. The eyes from dari depan sampai belakang, ya. most of the body is ice, so it's make this dragonfly is hard to catch, and they easily monitor the prey. Yeah, and this is the mouth part of the hemipteran predator. The mouth part is uh, short and sturdy, kuat dan kokoh. Ya, this is the example of the predator. This is Mermelontidae. Mermelontidae ini adalah undur-undur ya, ant lion. This is ant lion, adult of ant lion, carabid. Okay. Major types of prey searching behavior. Yeah, some of the strategies of the predator random strategies. So they move from another place to another place to searching the prey. It's different with directional. While the predator will move easily with directional cues by maybe from the sound, maybe from the shape of the body, maybe from the volatile compound that produced by the plant, and maybe from the, for example, from the chemical coating of the past, for example, that is directional move. Major type of prey searching behavior, that are three: sit and wait, trapping, and active searching. What differences sit and wait? Sometimes dragonfly and mantid. Dragonfly itu apa namanya? Dragonfly itu odonata itu ya. Ini dragonfly. And dragonfly and mantids, Manthodia mantida, they sit and wait. They sit, wait to catching the prey. They only sit and wait. And some of pests, they do trapping. They make trapping like ant lion Myrmelonthidae neuroptera. This is the larvae of the ant lion, of the undur-undur ya. This is the adult of ant lion. Yeah. The differences between this in with dragonfly is the antenna of Myrmelonthidae is longer ya, than odonata. And some of pests is active searching, like dragonfly, robberfly, sirpid fly. This is the larvae of the sirpid fly. This is predator. This is the prey, the aphid, and this is the adult of the sirpid fly. The adult is free living, hidup bebas, while the larvae phase is a predator. This is the beetle, Coleoptera, robberfly. This is many differences, and then after this we study about the biological control technique. The first one is augmentative. Augmentative is a breeding natural enemies and mass and releasing in plantation. So we do breeding. We develop the natural enemies in the laboratory, and after this we can release in a plantation. We can release in the ecosystem. It involve effort to increase population or beneficial effect of natural enemies. Various techniques we can do, yeah, to enhance the natural enemies, yeah, in the laboratory. And how to release in the ecosystem. We can do inoculative and we can do inundative. Inoculative, we release a small amount of the parasite of the natural enemies, yeah, while inundative we release many natural enemies in the ecosystem. What is the aim? What we expect by inundative and or release amount of or many natural enemies? The expectation of this inundative release, we release more natural enemies, is achieving a rapid reduction of the pest population. Yeah, this is how augmentative works. So we can artificially increase the number of biological control agents by breeding that natural enemies in the laboratory. The second one is conservative or conservation. So we do conserve natural enemies that already exist in the plantation. We can enhance the population of natural enemies in the plantation by reducing the adverse effect of pesticide. Yeah, we monitor how the frequency of the pesticide, how we do use correct dosage and concentration of pesticide, and uh, how we manage to use the synthetic pesticide. This is how we can reduce the adverse effect of pesticide. And then we can do habitat manipulation. We can enhance, for example, we can enhance the natural enemies, predator and parasitoid, by growing the plants that provide nectar, by growing refugia. And so we can produce nectar for the development of the adult parasitoid, and we can use using a straw mulch to protect predator. So from parasitoid they will get the benefit from pollen and nectar from the flowering plant. And then the third one is we can do classical biological control or introduction. We can bring natural enemies from overseas or and between island in Indonesia. If we didn't have a specific natural enemies of some pests, we can bring natural enemies from outside the country, for example. This is how in California works. They ever introduction Vedalia beetle, Rhodolia cardinalis, like this, to control the cottony cushion scale Itzeria burkasi. And in Indonesia, we also have invasive species uh, enter in 1986, Heteropsila cubana, kutu loncat lamtoro, and they enter Indonesia through Ciawi, Bogor, Indonesia, and we okay, and we can do control that Heteropsila cubana by bring natural enemies from the origin country of the pest. Jadi kita dapat mengimpor natural enemies dari negara asalnya. Karena hamanya ini kan hama invasif juga, jadi perlu kita belum punya musuh alami ya, salah satu case-nya seperti itu. And then this is the influence of flower refugia on biological control. Refugia, an area which population of organisms can survive through a period of unfavorable condition. So we do planting many flowering plant, for example, Tagetes erecta, sunflower, zinnia, and etc. What are the benefits? Benefit is support fitness due to nectar that has been produced by the plant. For adult parasitoid and predator, they can get the nectar. For its fitness, fitness itu adalah kebugaran ya, support fitness. Jadi before they able to searching the host or the prey in the field, they should fit before, so they will need the nectar and the pollen. Provide shelter for natural enemies. Yeah, they can also hide in this flowering plant. This is the flowering plant. This is a short explanation about invasive alien species. Is organism that introduced to an area where they are not known to occur. Yeah? So invasive species is a species that come from another country, for example. It could be a virus, bacteria, insect, plant, weed, or other organism. So invasive species or alien species, yeah, it can enter uh, one of the country uh, and it will threat our diversity, and also it will threat our crops, ya akan mengancam tanaman kita. What are the example? The example is Spodoptera frugiperda. They came from United States and they entered in Indonesia in 2019. And this is the egg mass of the army worm, Spodoptera frugiperda, Noctuidae, Lepidoptera, and this is the egg. And after they hatch, they will maybe 200 larvae will come out from this egg, and then the newly hatches larvae for armyworm. This is the mature larvae, and this Spodoptera frugipeda is a main pest of uh, sweet corn and maize in Indonesia. And now the Spodoptera frugipeda is spread all over Indonesia. This is why in a hat we can find Y shape Spodoptera frugiperda, and the damage they consuming the leaf tissues from the young larvae. They will leaving the opposite epidermal layer intact, yeah, and the late instar they will make big holes in the leaf. And what are the natural enemies? The natural enemies, we can use parasitoid for example, Cotesia like this, Cotesia and Chelonus. And also we can use numerous pathogen. As we know that pathogen is microorganism that can make some organism become ill, including virus, fungi, protozoa, nematoda, and other. This is the entomopathogenic fungi Nomorea relay. Yeah, this is Paracoccus marginatus. This is also entered Indonesia in 2008. Uh, the host plant is over 55 plants. They can attack papaya. They can attack many plant, mango, avocado, and etc. They are from Mexico, Costa Rica, Guatemala, Mexico, and the damage of the plant, uh, chlorosis. So the color is because this pest is suck because this pest is sucking the sap of the plant. So the maybe in the leaf there are distortion, stunting to kerdil, and maybe the fruit or the leaf become fall and production of honeydew. This is the apa ya? This is the Paracoccus marginatus, and they also produce honeydew atau embun madu. Like in aphid, they also produce a honeydew, and because of this honeydew, it okay, this honeydew is a good medium for the growth of sooty mold. So ini adalah dengan adanya embun madu itu merupakan media yang baik untuk untuk tumbuhnya embun jelaga, ya, sooty mold. The sooty mold is commonly the color is black, and possibly the death of the plant. The natural enemies of the papaya, like Cryptolaemus, lady beetle, lashwing, hoverflies, and etc. And after this, we can see the predator and parasitoid. This is the predator, Cryptolaemus montrosieri, mealybug predator. This is the lashwing Chrysopidae. The egg is so specific because this egg have like a thread, yeah, like a benang, yeah, like a thread. This egg is supported by the stalk or thread. And this is the larvae and adult of circuit fly. This is circuit fly, adult circuit fly. Adult of the circuit fly is a um, free living, while the larvae is they are parasitoid. Sorry, they are predator. They searching for the prey. And this is also the invasive species Pomacia caneliculata. They, they damage our rice plants. This is the egg, and the hatchability is about 90%. And the pinky one, and they are able to breathe in the mud. These golden snails originally from South America. This is become our main pest in paddy field. They enter in Indonesia in 1980. And this is I have one video that we can watch together. It's really nice. It's about the biological control technique. The first one is augmentative. Second one conservative, and the third one introduction. This is Pac-Man. The yellow one is pass. Okay, and this is the ghost. The ghost. This is the natural enemies. Natural enemies. Natural enemies could be predator or could be parasitoid. And how to manage the pest by augmentative, conservative, and introduction. So the first one is augmentative. We do artificially increase the number of biological control agents, yeah, by producing the natural enemies in the laboratory, and we can release into the plantation, so it will can control the pest. Then second strategy is conservation. Enhance the habitat of the biological agent by reducing the insecticide, or we can do planting refugia, so the biological enemies will breed easily and attract the pests. And the third one is the classical biological agent or introduction. We introduce to natural enemies from outside country, from the native habitat of the pest, to control the pest. So this is how augmentative and conservative introduction works. So I hope this video make you easier to study about biological control technique. Oh no, we finished all the section today. Don't forget you should do resume lectures and resume the video. Please attach the resume video in the lectures, in the resume lectures. Thank you very much. See you in Zoom. Wassalamualaikum warahmatullahi wabarakatuh. Bye bye.